If you conduct imaging experiments and measure multiple cellular parameters, then you know how overwhelming the resulting data sets can be. As powerful as microscopy is for generating stunning images, analytical power only comes from segmentation, image analysis, and careful multi-parametric data interpretation. Results come from powerful tools that review the image data and confirm the findings. You need to be able to view your data from large populations down to images of individual cells. You also need an easy way to clearly present your image data and the final results. The Novo software approach allows you to make the most of your image data by enabling population-based analysis and presentation of your image cytometry work. FCS Express 4 Image Cytometry has features designed specifically for image data analysis, presentation, and reporting. Our new imaging platform leverages proven population analysis strategies from flow cytometry with new image handling features that allow you to quickly import, manage, analyze, and display your imaging data. FCS Express 4 Image Cytometry builds upon the state-of-the-art analysis methods used in FCS Express 4 flow cytometry. Work with your image data to easily generate plots and present your results. Then create reports with the unparalleled presentation capabilities pioneered by Novo software, all within a modern and intuitive user interface similar to the Microsoft Office suite. Once images are segmented and multi-parametric data is generated, flow and image cytometry data are analyzed much the same way. The traditional concept of gating on plots to define cellular subpopulations, common in flow cytometry, has powerful implications for image cytometry. You can establish profiles for cells of interest and use image gates to view only the selected cells in image galleries, or go back and see them as highlighted in the original images. Our new software allows you to easily data mine and then interact with your multi-parametric data and the corresponding images, allowing you to easily explore and optimize your assays. FCS Express 4 Image Cytometry is the only software that provides all of these powerful tools in one package. FCS Express 4 Image Cytometry will help you manage and make sense of your data, quickly load an experiment's data into customized templates, adjust settings and filters for the new experiment, and when finished, export your completed reports in the appropriate format. Use FCS Express 4 Image Cytometry and spend your time doing more science, getting to your results faster, and generating high-quality presentations with a few clicks of a mouse. As always, FCS Express 4 Image Cytometry is compatible with the entire suite of Microsoft presentation tools. This means that a single click of a button can create PowerPoint presentations, Excel spreadsheets, and even PDF documents that look exactly like your analysis. We will now demonstrate the capabilities of FCS Express 4 Image Cytometry. Using a nuclear translocation dataset, we will optimize an assay, review images of cells in multiple conditions, run batch analysis, and create a final report. Our tools help you confidently move from assay development to routine analysis and production. Nuclear translocation is a common cell signaling event. Upon cell activation, signaling molecules present in the cell cytoplasm move or translocate to the nucleus, usually to modify gene expression. Activated cells change from having diffuse cytoplasmic staining to having bright, concentrated staining within the nucleus. With FCS Express 4 Image Cytometry, you can use flow plots and gating to help select the optimal image analysis parameters to follow for a robust and optimized assay. FCS Express 4 Image Cytometry lets you display your image analysis data in heat maps, single image picture plots, or dot plots and histograms for population-based analysis. Plot any of your extracted feature parameters on any axis of any plot. This is useful for visualizing the most valuable parameters to use when optimizing the sensitivity of an assay. Heat maps can be used to select particular wells of interest. In this case, we will select the positive and negative controls for the experiment based on the plate map. Gates from a heat map can be applied to dot plots to isolate your controls. Not every single cell responds identically in a positive control well, and in the negative controls, cells may auto-activate. Notice that the translocated cells make up greater than 90% in the positive control gate, but less than 10% in the negative control gate. This is typical but can now be accounted for. We use the tools in FCS Express 4 Image Cytometry to better understand the results at the single cell level. In this case, the control wells are behaving as expected. Cell galleries neatly display single cells excised from images based on your gates or filters. View galleries to visually confirm gating strategies. Easily find unusual outliers and adjust gating if required. Notice that as I move this gate, the gallery updates automatically. Image gating in FCS Express 4 Image Cytometry allows easy and real-time visualization of populations and statistics. Notice that as I move my well gate, the dot plot updates automatically to show only the cells within the selected well. You can also select data points on the dot plot to visualize the corresponding cells on a picture plot. In flow cytometry analysis, we call this backgating, and you can see how powerful it is when finding cells of similar morphology from within a mixed population using images. When you draw a gate on your dot plot, instead of only showing the cells in the gate, cells become highlighted in the gallery. Image gating allows the user to drill down to individual cells of interest in cell galleries, right next to relevant plots and statistics. You can also create a gate within the cell gallery and view only those cells in other plots. Plots and statistics from multiple wells can be overlaid or combined to explore the differences between experiment and control data. Batch processing can be used for analysis of single images, several images, or many images from multi-well plates in high-content screening for hit identification. During the batch process, you can automatically create PowerPoint presentations, PDF files, spreadsheets, or send data to your laboratory information system. Z-prime values can also be calculated directly in the layout and will update automatically as gate statistics update. Once your data analysis is complete, FCS Express makes publishing your results easy. Generate individual publication quality figures or export complete reports. In this demo of the image cytometry features, we have optimized an assay, reviewed images of cells in multiple conditions, run batch analysis, and created a customized report. Whether you are looking at single, several, or hundreds of images, FCS Express 4 Image Cytometry can automate the data analysis process to get you results in record time. FCS Express is trusted and used by thousands of researchers around the world to publish in the most prestigious journals. Be confident that FCS Express 4 Image Cytometry will manage your image cytometry data analysis and reporting as well. FCS Express runs on all current Microsoft Windows operating systems and can import data from multiple imaging platforms. More formats are enabled all the time. Check our website for current compatible formats. FCS Express improves assays, shortens your workflow, and automates time-consuming menial tasks. Can your other analysis packages do all of that? Spend your time doing what you do best, science. With FCS Express, no amount of data is too intimidating. See for yourself. Try a demo of FCS Express 4 Image Cytometry. For examples of the many advanced flow cytometry data analysis tools already in FCS Express, please see other videos available on our website. FCS Express 4 Image Cytometry, turning your image cytometry data into results.